Hi, I'm Jonas, and I'm a researcher here at the Center for Plant Systems Biology at the VIB. I work in the Thomas Jacobs lab, where I'm involved in a number of projects related to genome editing. Because I have to generate many different constructs, I spend a lot of time doing molecular cloning. Our go-to method for this is Golden Gate cloning, because it is very efficient, and there are many DNA building blocks already available for me to use. But there is one annoying feature, and that is all the different standards that are used. Let me explain why. Golden Gate cloning is a great method if you have the correct assembly standards at your disposal, but I've had too many occasions where my building blocks weren't compatible. Instead, we've developed the use of single-stranded DNA oligos with Gibson assembly to bypass the design standard problem in a process called Oligo stitching. We can actually use Oligo stitching for 4 different applications. 1. To convert Golden Gate parts between different assembly standards. 2. To directly assemble incompatible Golden Gate parts. 3. To generate de novo sequences without PCR amplification. And 4. To domesticate plasmids. In other words, to remove restriction enzyme recognition sites that interfere with Golden Gate cloning. This last application can also be extended to perform saturation mutagenesis. I'll briefly explain all 4 applications for you. Let's start with clone conversion. The first step of the process is a restriction enzyme digest. A donor and an acceptor plasmid are digested with the correct restriction enzymes, resulting in the desired factor backbone and Golden Gate part. The second step is the Gibson assembly. Here, the designed DNA oligos with homologous sequences are added to the reaction. The 5' exonuclease chews back the 5' ends so the oligos can anneal. Afterwards, the DNA polymerase fills up the nucleotide gaps and the added oligo becomes double-stranded. Due to the continuous activity of the 5' exonuclease in the single isothermal step, the 5' end of the now double-stranded oligo is degraded as well, resulting in annealing of the Golden Gate part. DNA polymerase again fills the gaps and DNA ligase seals the nicks. The result is the desired Golden Gate part in the desired backbone. You can play around with this concept by adding extra sequences to the oligo, like an extra stop codon, linker, or even larger sequences. The DNA polymerase proofreading activity even allows us to delete existing stop codons or other sequences in the Golden Gate part. If there is a mismatch between the oligo and the Golden Gate part, the DNA polymerase will start with a 3' chew back before polymerization. In an extension of clone conversion, oligo stitching can also be used for directly assembling incompatible parts. The first step of this process is again the restriction enzyme digest. 1 acceptor and multiple donor plasmids are digested with the right restriction enzymes, resulting in the desired factor backbone and Golden Gate parts. The second step is the Gibson assembly. Here, the designed DNA oligos with homologous sequences are added to the reaction. Extra oligos are added to merge the multiple desired Golden Gate parts or even PCR-derived sequences. The 5' exonuclease chews back the 5' ends so the oligos can anneal. Afterwards, the DNA polymerase fills up the nucleotide gaps and the added oligo becomes double-stranded. Due to the continuous activity of the 5' exonuclease in the single isothermal step, the 5' end of the now double-stranded oligo is degraded as well, resulting in annealing of the Golden Gate parts. DNA polymerase again fills the gaps, and DNA ligase will seal the nicks. The result is the desired Golden Gate parts merged in the backbone. A third application of oligo stitching is the Novo parts assembly. We use this method for generating prime editing guide RNAs, but this can be used to generate other sequences as well. The first step of this process is again the restriction enzyme digest. The acceptor plasmid is digested with the correct restriction enzymes, resulting in a linearized backbone. The second step is the Gibson assembly. Here, the designed DNA oligos with homologous sequences are added to the reaction. Several oligos can be merged together to create the desired insert. DNA polymerase fills up the nucleotide gaps and the added oligo becomes double-stranded. Due to the continuous activity of the 5' exonuclease in the single isothermal step, the 5' end of the now double-stranded oligo is degraded as well, so the oligos can anneal. Afterwards, the DNA polymerase fills up the nucleotide gaps and the DNA ligase seals the nicks. The result is the desired sequence in the backbone. Finally, oligostitching can be used for the removal of restriction enzyme sites, which is needed for part domestication. Another application is saturation metagenesis. In this application, we start with the desired factor containing a restriction enzyme recognition site that needs to be domesticated, or a region close to a restriction enzyme site that is to be mutagenized. After the restriction enzyme digest, two types of oligos can be added for the Gibson assembly. For domestication, an oligo with a silent mutation at the restriction enzyme site is added, while for saturation mutagenesis, a pool of oligos with degenerate bases to mutagenize one or more amino acids is added. The 5' exonuclease chews back the 5' ends of the vector backbone so the oligo can anneal. Afterwards, the DNA polymerase fills up the nucleotide gaps and the DNA ligase seals the nick. The result is a mutagenized part in the desired backbone. In short, we are trying to overcome the limitations of design standards. I have found that oligostitching is a very useful method for the 4 applications that I've described to you: clone conversion, incompatible parts assembly, de novo parts assembly, and metagenesis. With this video, I hope that oligostitching can help you in your molecular cloning projects as well. Happy cloning!